Hi everyone. In this class, we'll see about the protein metabolism. The objectives are introduction for the protein metabolism and transamination, deamination, fate of ammonia and transport of ammonia. So before going to the topic, we'll see just about the introduction of protein metabolism. Uh, the amino acids are essential to life. They have a role in metabolism and are important in nutrition. So, by these amino acids, the proteins are synthesized. So, they form short polymer chains called peptides, as well as longer chains that are called polypeptides or proteins. So, about 75% of the human body is made up of chains of amino acids, which is why they are so vital to how your body system functions. And all the chemical reactions that occur in the body depend on amino acids and the proteins they build in. And these amino acids are derivatives of carboxylic acids formed by substitution of alpha hydrogen for amino functional group. So in this picture we can see one end of the amino acid which is a carboxylic group present, and another end will be the amino acid. So there presents a alpha carbon. These two functional groups are attached to the alpha carbon of a amino acid. And these amino acids are digested by the proteases. So they are classified as exopeptidase and endopeptidase. So the enzyme hydrolyzes the peptide bond at the site of particular end. So here the proteolytic enzymes include endopeptidase and exopeptidase. So what is endopeptidase? They act on peptide bonds inside the protein molecule so that the protein becomes successfully smaller and smaller units. So this group includes pepsin, trypsin, chymotrypsin and elastase, whereas exopeptidase are which act only on the peptide bond located at the ends of the polypeptide chain. So under these exopeptidase, its carboxypeptidase and aminopeptidase are classified. So these carboxypeptidase which acts only on the peptide bonds at the carboxyl terminal end of the chain, and aminopeptidase which acts only on the peptide bond at the amino terminal end of the chain. So these amino acids are digested by enzymes and in stomach, pancreas and intestine. So in stomach by HCl and pepsin, in pancreas as trypsin, chymotrypsin and elastase enzyme, and in intestine carboxypeptidase, aminopeptidase and dipeptidase. So when we consume proteins, they get digested to amino acids. So in the stomach hydrochloric acid is present, as we call it as HCl. It makes the pH optimum for the action of pepsin and also activates pepsin. The acid also denatures the protein. So there will not be any digestion in the mouth. So it directly goes to the stomach. Thereby HCl, the protein are denatures. And the renin, otherwise called chymosin, is active in infants and is involved in the curdling of milk. So it's absent in adults. Only infants it's the enzyme which is present. And the pepsin, it's secreted by the chief cells of the stomach as inactive pepsinogen. The conversion of pepsinogen to pepsin is brought about only by the removal of the amino acids from the N-terminal end by the HCl, which is produced in the stomach. And the optimum pH for the activity of pepsin is around 2, and pepsin is an endopeptidase which catalyzes hydrolysis of the bonds formed by carboxyl group of phenyl, tyrosine, tryptophan and methionine. So when it enters to the pancreas, there the digestion of proteins also occurs, as the optimum pH for the activity of pancreatic enzymes as pH 8 is provided by the alkaline bile and pancreatic juice. So the secretion of pancreatic juice is stimulated by the peptide hormones cholecystokinin and pancreozymin. So these pancreatic juice contains the important endopeptidases, and namely trypsin, chymotrypsin, elastase and carboxypeptidase. So these enzymes are also secreted as zymogens, means their inactive forms as trypsinogen, chymotrypsinogen and proelastase, so that the pancreatic acinar cells are not autolyzed here. And all the three are serine proteases. So when it comes to trypsin, trypsinogen is activated by the enterokinase present on the intestinal microvillus membrane. So once activated, the trypsin activates even other enzyme molecules also, and trypsin is activated by the removal of a hexapeptidase from N-terminal end. This trypsin catalyzes the hydrolysis of bonds formed from carboxyl group of arginine and lysine amino acids. So here the acute pancreatitis means premature activation of a trypsinogen inside the pancreas itself will result in the autodigestion of the pancreatic cells. So this condition is called acute pancreatitis. And the chymotrypsin, which act on chymotrypsinogen, it's activated by the trypsin in such a manner that A, B and C peptides are formed. So these three segments are approximated so that the active site is formed, and the selective proteolysis produces the catalytic site. So next we'll see carboxypeptidases. Trypsin and chymotrypsin degrade the proteins into small peptides. These are further hydrolyzed into dipeptides and tripeptides by carboxypeptidases present in the pancreatic juice. So these carboxypeptidase enzymes, which acts only on the carboxyl end of the amino acid, and procarboxypeptidases is activated by trypsin. They are metalloenzymes requiring zinc. And the intestinal digestion of proteins, complete digestion of the small peptides to the level of amino acids is brought about by enzymes present in intestinal juice, that is succus entericus. And this luminal surface of intestinal epithelial cells contains the following enzymes, as amidopeptidases releases the N-terminal amino acids, and dipeptidases and tripeptidases will complete the digestion of the protein. So once these proteins are digested to form amino acids, there comes the absorption of amino acids role. The absorption of amino acids occurs mainly in the small intestine. It's an energy requiring process, and these transport systems are mediated and or ATP sodium dependent import systems. So L amino acids are actively transported across the intestinal mucosa, and D amino acids are transported by only passive diffusions. So once these amino acids are absorbed by the body, then it catabolized by the reactions which includes transamination, deamination and transdeamination, and the formation of ammonia. So here we'll come to the topic, that is transamination. It's a transfer of amino group from one amino acid to another keto acid with the formation of a new keto acid and new amino acid. So this reaction is catalyzed by specific transaminases. So it requires pyridoxal phosphate as a cofactor, and these are specific transaminases for different amino acids. Examples: transamination of alanine, aspartate, phenylalanine and tyrosine. And threonine and lysine do not undergo these transamination reactions. So transamination is more predominant in liver, kidney, heart and brain. So this is an example of transamination reaction by alanine aminotransferase, where glutamate gets converted to alpha ketoglutarate by this enzyme with the cofactor pyridoxal phosphate, where it also occurs the conversion of pyruvate to alanine. These reactions are reversible. And the biological significance of transamination reactions are even metabolic and clinical significance. As in metabolic, this is the removal and detoxification ammonia takes place, and aspartate aminotransferase, that is AST, and alanine aminotransferase, that is ALT, are induced by glucocorticoids, which favors gluconeogenesis. So biosynthesis of these non-essential amino acids are also taken by transamination reactions. So these are the metabolic significance of transamination reactions. And when it comes to clinical significance, the AST occurs in myocardial infarction, and also infective hepatitis, alcoholic cirrhosis, and muscular dystrophy, whereas ALT is increased in liver diseases. And the first step of catabolism, in the first step, the ammonia is removed and the carbon skeleton of the amino acid enters into the catabolic pathway. So synthesis of the non-essential amino acids are also takes place in transamination, and by means of transamination all non-essential amino acids can be synthesized by the body from keto acids available from other sources. So for example, pyruvate can be transaminated to synthesize alanine. So here similarly oxaloacetate produces aspartic acid, and alpha ketoglutarate is transaminated to form glutamic acid. So those amino acids which cannot be synthesized in this manner are therefore essential. They should be made available in the food. So these are the biological significance of transamination reactions. So when it comes to transdeamination, it includes transamination with deamination. So both it forms a transdeamination, where transdeamination in all tissues, but deamination will occur only in liver. So the oxidative deamination as of amino acid is the removal of ammonia coupled to oxidative process. So here, where in the transamination reaction the amino group of a amino acid is shifted, but in oxidative deamination the removal of ammonia is taken place. So examples will be oxidative deamination of glutamate, L amino acids, and D amino acids. So this is the reaction where the glutamate gets converted to alpha ketoglutarate by the enzyme glutamate dehydrogenase, where NAD gets converts to NADH plus H plus with the help of water. So this is the example for the oxidative deamination which occurs in liver. As only liver mitochondria contain glutamate dehydrogenase, which deaminates glutamate to alpha ketoglutarate and ammonia. So all amino acids are first transaminated to glutamate, which is then finally deaminated by the transdeamination reaction. Here the amino acids are deaminated at the rate of about 50 to 70 gram per day. So during the transamination reaction, the amino group of all other amino acids is funneled into glutamate. So hence the glutamate dehydrogenase enzyme reaction is the final reaction which removes the amino group of all amino acids. It needs NAD plus as a coenzyme. So it's an allosteric enzyme which is activated by ADP and inhibited by the GTP. So the hydrolysis of glutamine also yields the NH3 plus, but it occurs mainly in the kidney where the NH4 plus, that is ammonia, excretion is required for acid base regulation. So when it comes to non-oxidative deamination, dehydratase are the enzymes which act on hydroxy amino acids to remove ammonia from these steps, as serine will give rise to pyruvate where threonine gets converted to alpha ketobutyric acid. So desulfahydrase enzyme where cysteine undergoes deamination and simultaneously transsulfuration to form a pyruvate, and histidine also undergoes non-oxidative deamination to form urocanic acid catalyzed by the histidase enzyme. Here the ammonia may also produced by the degradation of purines and pyrimidines due to bacterial purification. So next, after the transamination and oxidative deamination and non-oxidative deamination, there comes a formation of ammonia. So the first step in the catabolism of amino acid is to remove the amino group as ammonia, and this is the major source of ammonia. So however, small quantities of the ammonia may also be formed from catabolism of purine and pyrimidine bases. This ammonia is very highly toxic, especially to the nervous system. So detoxification of ammonia is by conversion to urea and excretion through the urine. So after the formation of ammonia, there requires detoxification of ammonia to remove the toxic material. And some other minor pathways are there to produce ammonia, as histidine to uracanic acid and NH3 by histidase enzyme earlier we saw, and aspargine to aspartate by asparginase enzyme, and serine and threonine to pyruvate and alpha ketobutyrate respectively by the dehydratase. And ammonia is also released during the purine nucleotide cycle. And purine catabolism and pyrimidine catabolism. In the gut, NH3 is produced by bacterial metabolism, which reaches the liver through portal vein. And here the L-amino acid oxidases of flavoprotein also releases ammonia from amino acids. Ammonia may also be produced in the GIT tract by bacterial putrefaction. So these are the minor pathways where the ammonia is synthesized or formed. And also cysteine undergoes deamination and simultaneous transsulfuration to form a pyruvate by the enzyme desulfurhydrase. So ammonia may be formed in the body through minor reactions like oxidation of MAO, that is monoamine oxidase, and the detoxification of ammonia, which reduces production and ammonia trapping and also urea synthesis. So in this reaction we can see ammonia trapping as a glutamine, and the transport of ammonia, which requires from the tissues of production to the liver, occurs in three forms. So wherever the ammonia is formed in the cell, liver cells, it has to be transported. So in the form of glutamine, glutamate and ammonia. So as a glutamine, the formation of glutamine mainly occurs in liver, muscle and brain. So the reaction, glutamine is formed from glutamate in a reaction catalyzed by glutamine synthetase that requires ATP, and transport as enters blood and reaches the liver. So significance will be, it's a non-toxic storage and transport form of ammonia. And when it comes to glutamate, it contains ammonia that is trapped in the transamination reactions and enters the blood and reaches the liver. And ammonia, the concentration of ammonia is in the blood is 15 to 60 micromolar per liter. So the ammonia present in free form may arise either from direct oxidative deamination and non-oxidative deamination of amino acids. So when it comes to fate of ammonia, ammonia is utilized in 5 ways: as conversion to urea, formation of glutamine, and formation of aspargine, and also formation of glycine, and formation of glutamine from alpha ketoglutarate. So these are the 5 ways where the ammonia gets converted to urea. So further we'll see this conversion to urea in the urea cycle, as here the urea is a cyclic pathway in which ammonia is converted to urea. It's called cyclical pathway because ornithine is used in the second reaction and generated in the last reaction, and the glutamine is formed from ammonia. Glutamine is used for several reactions, and also amino sugar biosynthesis and NAD biosynthesis. So this is the fate of ammonia, where we can conclude that the ammonia, toxic ammonia, gets to non-toxic as urea, glutamine or the glycine and all. So in the next class we'll see about the urea cycle and the reactions involved in urea cycle and the disorders involved it. Thank you.